Well, good morning, good afternoon, good evening, or late into the night, depending on which part of the world you are joining us. This is the symposium by the Oxford Longevity Project. I am Professor Denis Noble, one of the founders of the project, an emeritus professor of physiology in the University of Oxford, and I'm joined in chairing this meeting by Sir Christopher Ball, who was the head of one of the major colleges in the University of Oxford and is very well known for his public appearances and all kinds of things. And I think, Sir Christopher, you're going to say a few words before we launch into the meeting itself.
Thank you very much, Dennis. Yes, indeed. I think I'm here, you might say, under false pretenses, representing the patient. You've been kind not to mention my age, but I'm 86 years old. And about a dozen years ago, to my surprise, I needed a heart operation. It was a triple heart bypass. And I made a full recovery thanks to the wonderful treatment in the John Radcliffe Hospital, for which I shall forever be grateful. It's made me think about the difference between the doctor's responsibility and the patient's responsibility for our health as we grow through life. And I see it as a partnership. I'm sure many people do. But the question I often ask myself, and I want to hear answers to that today, is what should the patient be doing to ensure heart health throughout the life of a human being? My own answer to that question very quickly runs from the letters A to E. I have learned to avoid things that do me harm, like alcohol and beef and cigarettes. I've learned the importance of breathing, nasal breathing, and I tape my lips together at night to ensure that I breathe nasally over the night. C stands for cold showers every morning. D is diet. I eat a plant-based diet and avoid processed foods. And E is exercise, and I have a daily routine. I'm sure many people on this Zoom call will have their own routine. I hope to add to my A to E list something beginning with F and G today, and I shall be listening out for it. Because the question is, what else should we be doing? And I hope these remarkable experts we're going to hear from will help us do that. Thank you very much for inviting me to be here.
Well, thank you, Sir Christopher. We are now about to launch into the meeting itself, and I will be responsible for doing whatever bit of translation of technical terminology and so on might be necessary at the end of the presentations. Our first presentation is by one of the first groups in the world to show the functional significance of autophagy in the heart in an article published in a premier journal in 2007. And this is Professor Sadoshima. And Professor Sadoshima has been leading the field of autophagy in the heart through many publications and meeting presentations at international meetings. He's an associate editor of Autophagy and has organized many symposia. So, Professor Sadoshima, we're looking forward to your presentation to the Oxford Longevity Project Symposium. I hand the microphone over to you.
Great. So thank you very much, Dr. Noble, for introduction and also invitation to this society giving a talk. So I used to be a cardiologist in Japan many years ago, and I saw many heart failure patients, and that drives me conducting research. And I move into basic science and then still doing study regarding heart failure. And obviously, autophagy is one of the very important mechanism determining the fate of heart failure patient. So today, I would like to give you a very brief introduction about the role of autophagy in the heart. And autophagy is a major mechanism of cellular degradation through lysosome, and it is characterized by presence of double membrane structure, autophagosome. And the autophagy in cardiac muscle is very important because the cardiac muscle is hard to divide. You know, once you are born with your cardiac muscle, you know, cardiomyocyte rarely divide. So once you have some aggregate in a cell, it's hard to remove. So I believe that autophagy mechanism is particularly important for terminally differentiated cells like cardiac muscle. And we became interested in autophagy by studying the mechanism of myocardial hibernation. And hibernation is a major, amazing mechanism of cardiac muscle to survive, even in the presence of very severe ischemia. Basically, they stop beating and then conserve energy and then survive until doctor reopened the coronary artery. And we became interested in this amazing mechanism. So we had an animal model and we conducted unbiased study and found out that the autophagy is one of the major mechanisms activated in hibernating area of the myocardium. And we saw autophagosome, as you see here. And we found that in an area which has many autophagosome has less myocardial cell death, and area which has less autophagy has more cell death. So although this was a descriptive study, but we speculated autophagy supply energy to the muscle and also eliminate toxic material from this area. And it turned out that autophagy does many good things for the heart. And one of the most important one is the protection against the heart failure. And this is a mouse heart, and we can create a coronary ligation, and we can create ischemic area in the mouse heart, very tiny heart. And then if you leave this ligation for a while, we can create myocardial infarction. And we can determine which area of the heart activate autophagy by indicator mice developed by Dr. Mizushima. And we found that autophagy is activated in between infarcted area and survived area. And if you eliminate this autophagy by genetically altered mouse, we saw infarction is expanded, as you see here. And we saw more greater size of infarction. And also you can stain protein aggregate, and we saw more aggregate in autophagy loss of function mouse model. And also more importantly, those mice die more frequently than control mice. So these are probably sufficient evidence to say that autophagy protect the heart against heart failure. But the reality, even in the presence of autophagy activation, a patient eventually develop heart failure. So we believe that the autophagy is protective, but not enough. And we predict the presence of some mechanism to negatively regulate autophagy. And we studied intensively what this mechanism is. And we found one of the molecule called MST1 is quite important. And this is a component of the Hippo signaling pathway and promote cell death. But we also found that this MST1 inhibit autophagy. So in response to stress, MST is activated, not only kill the cardiomyocyte, but also inhibit autophagy, and that both of them cause cardiac dysfunction. And if you express this MST1 in cardiomyocyte, we saw almost complete disappearance of autophagosome. You see here, this dot is a reflector autophagosome, and we saw complete disappearance. And the mechanism is MST phosphorylate becrin-1. It's a very important molecule for the induction of autophagy. And this actually phosphorylation allows protein-protein interaction, electrostatic interaction with the BCL2. And the BCL2 is known to very potently inhibit action of a becrin-1, so that in the presence of MST1, becrin-1 binds to BCL2 and autophagy is inhibited. And to show the functional significance, we made a mice that MST1 is inactivated. And you see here, survival rate is dramatically improved. Mice in which MST is inactivated, they don't die. Survival rate is much less. But if you eliminate autophagy function in these mice by crossing these mice with mice without autophagy, this survival advantage disappear. So we believe that this survival advantage is caused by activation of autophagy. And same thing happens in human. And I have a former fellow in Taiwan who does cardiac transplantation. So he gave me heart failure heart and normal heart. And we saw MST is activated in failing heart. And the becrin-1 phosphorylation is increased, and autophagosome formation is inhibited. So this indicates that autophagy MST1 regulation is critical. So this becrin-1 BCL2 interaction can be targeted for the treatment of heart failure. And if you can invent a method to dissociate this becrin-1 BCL2, autophagy is activated. And also BCL2 is known to interact with apoptosis cell death molecule, so that this interaction can prevent cell death in the heart. So I strongly believe that this is an important target of heart failure treatment. And the lack of autophagy is also involved in aging. And many researchers has already reported that aging activates some mechanism to prevent autophagy. So by age, autophagy activity gradually decreases. But if you restore this autophagy mechanism by using genetically altered mouse model, in this case, Dr. Bess Levin's mouse, lifespan longevity is restored, increased, so that this suggests the functional significance of autophagy in aging. And we recently looked at the effect of autophagy suppression upon senescence. And senescence is a very special condition of the cell, which can survive even in the presence of stress. But not only that, it secretes some factors so that senescent cells make surrounding cells damaged. And also a senescence factor itself can induce senescence in other cell type, other surrounding cells. So we used the mice that in which autophagy is eliminated in the heart. And we saw many markers of senescence such as cell cycle inhibitors and cytokines and DNA damage markers and inflammasome are all activated. So we saw a connection between autophagy suppression and aging. And as I told you that once senescence is developed, senescent cells secrete factors that make surrounding cell also make senescence. So this actually causes kind of a chain reaction. So people speculate that once we have a method to eliminate the senescent cells, we can eliminate aging process. And one of the way to eliminate the senescent cell is to inhibit the BCL2 cell cycle, cell survival factor promoter. And if you treat the compound which prevents survival of senescent cells, we can rejuvenate the animals. So we treated this autophagy deficient mice with this ABT compound and look at marker of senescence, in this case, a DNA damage marker, gamma H2AX. And we saw this DNA damage markers are increased in this ATG7 knockout mice, but by the treatment of senolytics, we can eliminate these senescent cells. And we saw decreases in cardiac contraction in loss of autophagy function mouse model, but by treatment of this with ABT, cardiac function was restored. So we believe that this promotion of senescence is important for suppression of autophagy to induce aging. And although autophagy is relatively non-specific, but the autophagy can sometimes specifically degrade some proteins and organelles. And when autophagy specifically target mitochondria, we call it mitophagy. And this mitophagy is particularly important for the heart because heart muscle is very enriched by mitochondria. Mitochondria is an intracellular organelle to generate ATP. So more than 30% of the volume of cardiac muscle is occupied by this mitochondria. And the mitochondria is densely packed in between muscle fibers. So when this mitochondria is damaged, it creates very produce toxic materials so that it can directly affect the contractility of cardiac muscle. And when mitochondria is damaged, mitochondria damaged portion was selectively sequestrated by autophagosome. There are two ways to selectively recognize damaged portion of the mitochondria. One is ubiquitin-dependent mechanism, and one is a receptor-dependent mechanism. And we examined whether mitophagy is activated by stress. So in this case, we created aortic constriction in mice. We create a ligation by operating mice. And heart is subjected to very high blood pressure. So heart is under very severe stress. And we found that mitophagy can be activated, but unfortunately, mitophagy activation is very, very transient. And we looked at the mitochondria dysfunction. We found that once mitophagy is inactivated, mitophagy starts to show dysfunction. So this mitophagy is kind of a adaptive protective mechanism, but it is not enough. And once mitochondrial dysfunction develops, we start to see cardiac contraction, it decreases. So there's a clear correlation between activity of mitophagy and mitochondria function in the heart. So then if you can reactivate the mitophagy, we can possibly prevent, delay the process of heart failure. And so far, most efficient method to activate the mitophagy is the TAT-BECLIN-1. This is a very short peptide, but it has developed by Dr. Veslevin's group, but it has the ability to mobilize autophagy regulators from intracellular storage site. And we saw TAT-BECLIN-1 treatment reactivate mitophagy in cardiac muscle. And we found that if you give this TAT-BECLIN-1 in pressure overload mice, heart failure was prevented. But this restoration was not observed in autophagy deficient mice, so that we can say that this TAT-BECLIN-1 rescue cardiac dysfunction by activating mitophagy, you see here. But very interestingly, we found that general autophagy, this is not a specific form of autophagy, and mitophagy occurs in different time course in mouse heart. And this is really puzzling because mitochondria autophagy and autophagy should be mediated by same mechanism. Autophagosome formation mechanism should be the same, but the time course is different, and the mitophagy is activated when general autophagy is inhibited. And same thing happens in obesity cardiomyopathy. So if you have metabolic disorders such as obesity and type 2 diabetes, the many patients develop cardiac dysfunction. So we fed the mice with high-fat diet. 60% is saturated fatty acid, and look at autophagy is activated, and as expected, 6 weeks high-fat feeding activate autophagy. But this doesn't last long, and by 20 weeks, autophagy come back to baseline level, and FLAX is no longer activated, and mitochondria no LC3-2. But if we examine mitophagy using different markers, mitophagy continue to be activated even after 20 weeks, and we confirmed that presence of autophagosome by electron microscopy, and this mitochondria is sometimes degraded. So we are very confident that even at the time autophagy is completely inactive, mitophagy can be activated. This is probably because mitochondria is such an important organelle for cardiac muscle. So we speculated some compensatory mechanism, pinch hitter mechanism is activated. And we found that instead of conventional mechanism, alternative mechanism can also sequestrate damaged mitochondria and degrade mitochondria. And although I said this is just a pinch hitter, but in terms of disease, many diseases are chronic, like diabetes is a chronic disease. And it turned out that this pinch hitter mechanism is more important for the chronic disease because conventional mechanism is activated, but it doesn't last long. But the pinch hitter mechanism try to compensate the loss of conventional mechanism. And in fact, that if we inhibit this pinch hitter mechanism, cardiomyopathy was exacerbated. And also, if you look at the mitochondria become very scarce. This is a crystal structure is disorganized, and the heart to start to accumulate more lipid. So we believe that studying the molecular mechanism of this pinch hitter is as important as studying the conventional mechanism autophagy and mitophagy. So now I switch gear. I told you many good things about autophagy and mitophagy, but like many cellular responses, when the function is dysregulated, it can be harmful. And it is true also in the case of autophagy. And it turned out that when autophagy is activated too much, it causes cell death. And one of the forms of cell death is autosis. And autosis is characterized by morphological feature and biochemical features. One is the presence of many vacuoles, and also very characteristic space next to the nucleus. This is a cartoon, this space, we call it the perinuclear space. And this is a signature of autosis. And also autosis can be inhibited by cardiac glycoside, digitalis. It's an inhibitor of sodium potassium ATPase. And we found that this autosis is activated in reperfusion heart. And this is a mouse heart again, but if you create ischemia, and then if you remove this tie, this area becomes perfused again. And although having reperfusion is good in terms of oxygen and nutrient supply, but it causes very severe oxidative stress. And in fact, that cardiomyocytes die injured very severely by this reperfusion. And if you follow up the time course after reperfusion, late phase, after 6 to 24 hours, we saw many signs of autosis, like vacuole formation and perinuclear space. And we are excited about this finding because many people believe that cardiomyocytes die within first few hours. Within 1 to 2 hours, most of the infarction is done. But we found that myocytes continue to die using different mechanism even after 6 hours. And to getting more convincing proof that this is autosis, we treated the mice with a cardiac glycoside because by definition, autosis should be inhibited by cardiac glycoside. But the problem is mouse heart is not so sensitive to cardiac glycoside. So Dr. Bess-Levine and the Duke Foundation mechanism, we got the humanized sodium potassium ATPase mice so that these mice, sodium potassium ATPase is sensitive to cardiac glycoside. And we treated the mice with wobine, one of the cardiac glycoside. We saw autosis is prevented, you see here. And at the same time, myocardial infarction is also reduced. So this indicates that autosis is involved in myocardial injury, but cardiac glycoside dependent autosis inhibition can be considered for the treatment of cardiac myocardial injury. Since autosis is observed at the late phase of reperfusion, we are curious what's happening autophagy in late phase. And autophagy is a kind of a multiple step mechanism. One initial phase, autophagosome is formed, but that autophagosome must be degraded at the lysosome. So, if any step is inhibited, autophagy cannot be executed. So, in order to examine this series of flux is activated or inhibited, we use a special mice developed by Japanese group. And we found that in the early phase, autophagy flux is activated, but the late phase, autophagy flux is stopped in the middle somehow. So we examined various mechanisms, and we found that two molecules are particularly important at the late phase. And one is Rubicon, another is TFEB. And we found Rubicon is upregulated late phase, and the TFEB gradually time-dependently upregulated. So what's going to happen if we have upregulation of these two molecules? So Rubicon is known to prevent fusion between autophagosome and lysosome, whereas TFEB promote formation of autophagosome. So when this stop is inhibited, autophagosome kept generating. And also, in order to make autophagosome, cells have to use intracellular materials, and this material is known to be recycled. So once this autophagosome moves to lysosome, this is degraded, and this material can be used for the generation of autophagosome. So when Rubicon stops this process, this recycle is also inhibited. So as a consequence, we believe that the more autophagosome accumulates, whereas the material to make autophagosome is depleted. So to test this hypothesis, we use the Rubicon knockout mice, and we are able to show that the autophagosome formation accumulation is actually inhibited, and also characteristic feature of autosis is inhibited, and we saw decreases in myocardial infarction size by 20%. So we believe that this mechanism is important. So when autophagy is dysregulated, sometimes autophagy brutally eliminates many things in cells, and this is certainly pathological. And we know that it happens sometimes during the organ development. But another form of autophagy dysregulation is a kind of a mismatch between formation and degradation, and that allows accumulation of autophagosome, and this is not very good for cells. So to summarize my today's talk, I have only 3 slides, and I believe that autophagy is very important to maintain cellular homeostasis, and this is particularly important for terminally differentiated cell like cardiomyocyte. But unfortunately, this autophagy is sometimes not enough or even actively inactivated. And we know this happens in heart failure, high blood pressure, diabetes, and aging. And this creates a condition in which autophagy is insufficient, so that we have to find out the mechanism and try to restore it. And sometimes autophagy is dysregulated, and we believe that this happens in reperfusion and acute myocardial infarction, lysosomal storage disease. Unfortunately, I didn't have a time to show you evidence, but in this case, we try to remove this excessive mechanism and try to normalize autophagy mechanism. And finally, I just want to conclude my talk by introducing recent Nature paper showing that intermittent time-restricted feeding activates autophagy and prolonged lifespan in Drosophila. And the idea is that autophagy is known to be activated in circadian manner, specific manner, and it's known that the component of autophagy such as ATG1 and ATG8 are activated during nighttime. Usually, animals do not eat at nighttime, I guess. And so idea is by creating a very prolonged fasting time during night, we can stimulate this circadian mechanism to activate autophagy periodically every other day. And although this was conducted in Drosophila and also not specifically talk about the heart, but I believe that this is very relevant for the heart, because it's well known that the heart disease occurs in circadian manner, and particularly prime time for myocardial infarction is the early morning. So it's not a bad idea to have a prolonged fasting time during night. And in fact, that my friend, Dr. Diwan in WashU, reported that intermittent fasting is very good in preventing ischemia reperfusion injury. And he pointed out many beneficial effects for the heart. And of course, intermittent fasting is good, but it's tough, particularly every other day, 20 hours, almost impossible and even harmful for the health. So of course, any drastic intervention is not good, should not be good. But, if you consult to your doctor and then find a way to periodically clean up your heart, I think it may not be a bad idea to prevent the occurrence of ischemic heart disease and other chronic heart disease, heart failure, and also metabolic syndrome mediated cardiomyopathy. So with that, I'd like to conclude my talk. I'm thanking my fellow, and also I just want to give a special thanks to Dr. Beth Levine, who passed away two years ago, but she gave us a tremendous help to our lab and our autophagy research could not be conducted without the help of Dr. Beth Levine. And finally, if you have any question or idea of a collaboration, please contact me to this email address. Thank you very much for your attention.
Sorry about that. Thank you very much, Professor Sadoshima, for absolutely cutting-edge account. There were some technical terms in your presentation. I thought that before I ask Christopher whether he has a question for you, some of the chemical compounds in the body that you found have very big effects, like MST1, Rubicon. Those are not themselves drugs that people can buy, are they? These are the chemicals that the body is using in one way or another. I thought I should clarify that for the lay audience.
Yeah, so what I showed is a very basic preclinical evidence. So kind
Exactly.
of only proof of concept. And I know some people developed MST1 chemical inhibitor, Chinese group. I saw some publication and we have not tested, but if we have more evidence, like a large animal study or something, I think it's interesting to develop compound to do the same thing as loss of function in mice, and but we still need to work on it.
Exactly, that is the role of fundamental research,
yes,
which you're doing, which is to point the way to pharmaceutical companies and others who might develop drugs that do the same kind of thing. Because what you showed, particularly with MST1, was very impressive change in longevity, really
yes.
Quite remarkable. So Christopher, do you have a clarification point you'd like to put before we move on to Professor Choudhury's presentation?
Thank you very much. I take home from this wonderful presentation, for which I'm very grateful, 3 messages for the patient. And any indication of how we can achieve these three things that I think you've told us we need to do would be helpful. You've persuaded me that autophagy is a process that's very important to us. What can we do as patients to promote it? Question one. You've explained the effect of stress. And I suppose most of us know that we should try to avoid stress, but clearly I shall go away even more keen to avoid stress because of what you've told us. And the third point is the overnight fasting, which is fascinating. What sort of practical steps can we ordinary patients take to achieve the effect that you've discovered?
So in terms of how to stimulate autophagy, I think best way is to reduce the calorie intake. Like, Dr. Guido Kromer's group said that, metabolite, acetyl-CoA inhibit autophagy. So, you certainly animal model, the best way to induce, our lab, when we want to make a positive control of autophagy induction, we give a starvation, fasting to animals. So in general, caloric restriction is effective for many things. So rather than taking like a medicine which may have side effect, reducing calorie intake is important. Also, at the end, I talk about circadian activation, and it turned out that many of the autophagy genes are regulated in circadian manner, and it's known that nighttime is a prime time for the activation. So I think it's intermittent time restricted feeding is the same thing, but it's important to facilitate this process is good. And the other thing, we know that NAD is another mechanism to facilitate autophagy, and NAD is kind of a metabolite of the niacin and other things, so probably sorry to give a little bit the specific thing, but think about diet to increase the level of NAD may stimulate autophagy. And in terms of stress, yeah, I think mental stress is, of course, that we can easily imagine when you say stress, but I'm talking about high blood pressure and also metabolic stress. So try to reduce the blood pressure and restrict salt kind of things, and also diet, overeating and high carbohydrate probably is not good for the heart. So today I talk about ischemia, high blood pressure, metabolic stress, and all related to blood pressure and diet kind of things. So and the last three, in terms of intermittent fasting, I try to do it, but it's very difficult. And I know I shouldn't eat at night, but I start drinking and eating at night, and not good. But maybe start from overnight fasting is good, and always you have to talk to your doctor because basically what you eat and how much you eat dramatically affect various things in your body. So that's my comment.
Thank you.
Yes, thank you very much. It's good to know that many of the good old-fashioned lifestyle advice can be very important without having to necessarily wait for you and your colleagues to develop yet another new drug. I've got a question that's come through on the Q&A from one of the listeners, but I'm going to delay that until we come to the final discussion after Professor Choudhury's talk, because we really must move on now to Professor Choudhury. Professor Choudhury is Professor of Cardiovascular Medicine in Oxford. And I've got the wrong sheet. Here we are. Yes, he's going to tell us about an area that's also extremely important in relation to longevity, which is in his title, what happens when you look at immunity in macrophages. And so I'll hand over directly to you, Professor Choudhury, to give your talk, and then we'll go straight into discussion and further clarification. Over to you.
Great, thank you, Professor Noble. So I want to start really by fulfilling Sir Christopher's wish. So if we take, he was looking for an F and a G, and I'm going to put to him that he's got the F in autophagy. I realize that's a bit of a stretch, but I'm gonna claim it. And I now want to give him a G, in fact, two Gs, one in glucose and one in genes. And I'm gonna introduce some concepts today, that some of which might be familiar and some of which might be new. I'm going to try to relate them to my practice as an interventional cardiologist treating patients with heart attack and angina. Okay, so this is, I think it's a shame it's a Zoom meeting, because if it were not, I would ask you what it is. But I guess I'll have to tell you that this is an architectural blueprint. And it happens to be a blueprint for the Guggenheim Museum in New York, which you'll probably recognize instantly. Now I've pointed it out. But there's a reason that I'm showing you a blueprint. And that reason is that the blueprint is very relevant to consideration of planning in biological systems. So the blueprint allows for a process of more or less unlimited reproduction. It allows rapid and very faithful, accurate production of copies. It provides a negative of the original, that's hence it's white on blue. And interestingly, it doesn't allow reproduction of shades of color. It is very much a binary, white on blue. And of course, you've probably guessed already that I'm building up to talking about the blueprint for life, which is arguably the genetic code. Can you see my cursor or not? Dennis, can you see the cursor? Yes. Or maybe I need a laser pointer. Oops.
Yes, we can see your cursor, no problem.
Okay. Now I have a laser pointer.
You have a laser pointer.
Much more elegant. Okay.
Very clever. Yes.
So these are, of course, you'll recognize immediately the double helix of DNA, which contains the blueprint for life, contained within the sequences of these 4 bases shown here as these chemical structures. So they encode the particular building blocks that will make up the structure of interest. But it immediately brings about a problem, because every cell in the body contains the blueprint in its entirety. And the problem, therefore, that has to be overcome is in determining which are the genes that are expressed, the genes being the codes, to allow articulation of the particular building materials, in the cellular sense, the proteins, that will determine the structure and function of each of these different cells. And the long and short of it is that one way of doing this is through something called epigenetics, which in a textual sense might be a bookmark or a highlighter or underlining. It's what allows the emphasis on particular parts of the code so that those parts of the code can be translated into the proteins that go on to make up the cell and determine the cellular function. Okay, so that's important background that I want to get across, and we'll come back to that in a moment. So inside the nucleus of the cell, every single cell is DNA, but the epigenetic markers, so-called, these little bookmarks shown here as these little dots, are the things that determine whether the DNA is in an open or closed configuration. When it's closed, it can't be read, but just like a book, when it's open, it can. Okay, so that's the role of the epigenetics. Now, the disease that I want to talk about is heart attack or myocardial infarction. So here is obviously a heart. This is a coronary artery, of which there are three main arteries. And these coronary arteries, as we age, have a propensity to develop within their walls this material that we term atherosclerosis. And we're going to look at that in a bit more detail in a minute. But that atherosclerosis can narrow down and can even occlude, can block off the artery, such that if it does, excuse me, the muscle downstream of the narrowing can become damaged. And Professor Sadoshima was speaking to us a few moments ago about myocardial infarction. All we mean by myocardial infarction is that the damage, the injury that's inflicted upon the heart through interruption of the blood supply. And that still is the most common cause of death in Western countries. So if we look at what this means in a clinical sense, this is the patient that's having a heart attack. Here is the coronary artery that we saw a moment ago, and you'll see it just comes to an abrupt end. There's no blackness, which is the X-ray dye that I put into the artery, going beyond this point. Then we put a wire down the artery. This is a little device on the end of the wire. And on that wire, we can thread balloons and small pieces of metal called stents to restore the openness, the patency of the artery, and to improve the blood supply to the heart at the other end. If we take this little device out of the artery, it's like a little windsock, you can see some of the material that causes heart attack. So this yellow friable material here is the atherosclerotic debris. It's cholesterol rich. It contains a lot of inflammatory material, and it was originally contained within the vessel wall. We'll look at some microscopic images of that in a second. This red material is red blood cells, the blood itself that's become trapped in some of the clot material and other debris that was occluding, that was blocking off the artery. And if we look in high resolution, you can see on the outside of this are some platelets. These are part of the blood coagulation system. So I like this slide because it shows all of the important components of atherosclerosis and the principal complication, myocardial infarction. Now, the disease that I really want to focus on, having given you these two pieces of background, so one on genetics and epigenetics and the other on myocardial infarction and narrowing of coronary arteries, is the role of diabetes. And we have recently undertaken some work which we think addresses an important paradox in diabetes, which is also important potentially to aging and to metabolic health. So what you're seeing here in the top is some material that's been extracted from a patient's coronary artery. And the panel on the left, panel A, is from a patient with diabetes. and on panel B is a patient without diabetes. And this brown mottling here is a stain for a certain type of cell called a macrophage, which is very important in the development of atherosclerosis. And it turns out that patients with diabetes, as shown in the graph, tend to have much more macrophage-rich atherosclerotic plaque than do their non-diabetic counterparts. Now, this for us was the beginning of an opportunity to address an important question. So diabetes, as you well know, is extremely common in the West and is expanding rapidly globally. And the thing about diabetes in relation to heart disease is that it confers a very substantially increased risk, both of having a first heart attack and of failing to survive it in the longer term once you've had that heart attack. And that I won't go into the detail, but that's more or less what this graph shows. Now, the paradox that I referred to is shown here. We know that whether you have type 1 diabetes or type 2 diabetes, the sugar itself, the elevated sugar, which is characteristic of diabetes, is an important driver of the disease process. But here's the rub. If you treat the glucose very intensively, and that's shown on this graph here, even for a period of several years, the risk of heart attack is not reduced by intensive glucose lowering. Okay, so that might come as a surprise. Glucose in diabetes seems to drive heart disease risk, but treating glucose does not correct that risk. And we ask the question, why might that be the case? And this is where the earlier slides that I've shown you now come back into focus, because we started to ask the question as to whether high levels of glucose might fundamentally alter cellular metabolism in a way that might change the epigenetics of inflammatory cells, the macrophages, and might change them in a way that led them to remember their previous exposure to high glucose, such that when the glucose was corrected, that mark, that imprint, that memory remained, and correcting the glucose alone would not therefore correct the harm that had been done to the inflammatory cell, the macrophage. So in order to test this in the first instance, again, we also went to mice, and we're going to show you some slides from a scientific paper now, but I'll try and explain it as I go. So we took bone marrow from mice that had either not had diabetes or had diabetes, and we took out these cells, the macrophages that I showed you a few moments ago, and then we gave them some chemicals that would push them either to an inflammatory type macrophage or a different chemical that would push this macrophage to something that's more important in repair. And the long and short of it is that if you take bone marrow from mice that had diabetes, even after you return that bone marrow to normal glucose for a prolonged period, those cells show a propensity to become pro-inflammatory, harmful, and a resistance to becoming reparative. So these, if you like, would be conditions that would be important in driving the processes of atherosclerosis that we saw earlier. And indeed, we can do some more sophisticated tests. This is a very simple representation of quite a complex experiment. So here we're looking at gene expression within macrophages, and we look at all approximately 20,000 genes at a time, and we look and see which ones have been pushed up and which ones have been pushed down. And if we look in the basal state, you can see there are already distinct differences between diabetic origin cells and non-diabetic origin cells. So in other words, the expression patterns across these 20,000 genes are already different. But if we give those cells a little tweak with an inflammatory chemical, you can see that the differences are even more pronounced. So again, reinforcing the idea that diabetes is driving a pro-inflammatory state. Then we wanted to ask, whether that mattered and whether it mattered to our most important disease, atherosclerosis, which causes the coronary artery disease and myocardial infarction that I showed you at the beginning. And to do this, we also did a mouse experiment, and we took mice that were, excuse me, had a propensity to develop atherosclerosis. And then we transplanted into those mice bone marrow, either from controls or from mice that had diabetes. We then left those mice for fully 3 months so that the cells, even from the diabetic mice, were now in a completely normal glucose environment. It had all been corrected. It was akin to having perfect medical management of diabetes. And then we looked after 3 months at the degree of atherosclerosis in the conventional place in these mice. And again, without belaboring the point, I can tell you that the mice that received diabetic donor bone marrow had much more atherosclerosis than the mice that received the control bone marrow. So in other words, even putting the cells into an entirely normal glucose environment, there was a memory that they had previously come from a diabetic environment. This is a relatively complex slide, which I'm going to just bring your attention to panel C. These little labels here, H3K27AC and H3K4ME3, reflect those little circles and triangles that I showed you earlier, decorating the genetic material within the nucleus of the cell. And these are the bookmarks. These are the little markers that indicate which bit of the genome should be read within a given cell. And suffice it to say that when we do these sorts of plots, there's a very marked difference between the little bookmarks that we see in the diabetic mice and in the control mice. And importantly, what's shown in the bottom is that whilst those marks start off in the bone marrow, they are retained as those progenitor cells undergo differentiation and subsequent stimulation. So this is a robust and lasting programming of cells which originated from the high glucose environment. Coming to the end now, the data that I've shown you thus far have been for mice, but of course we really want to know whether or not a similar phenomenon might occur in patients. And I can tell you that we've done some experiments looking at human atherosclerosis, which indeed suggests that might be the case. But even more excitingly for us, if we look at peripheral blood cells, and we take blood cells from control donors or from patients with diabetes, we see very markedly different patterns of gene expression, even after we've rested the diabetic origin cells in normal glucose. There are about these red dots represents a gene which is differently expressed in the diabetic, statistically significantly so, versus the control. And there are about 3,000 of those. So very profound differences between peripheral blood cells in diabetes and control. So where does that take us? Why do we care? Well, we care for the first, in the first instance, because we think this might be one of the factors that addresses this long-standing paradox. We think the glucose is getting in to the inflammatory cells and their precursors and causing lasting effects. And that might have implications for the way we think about diabetes. At the moment, we regard it very much as a metabolic disease, but this sort of work would imply that there is an immune or a homeostatic, the mechanisms that maintain normal regulation of normal processes might also be affected. And that has knock-on implications for diagnosis and for monitoring of treatment, but also, of course, for making opportunities to treat patients with diabetes. And one of the things that we're particularly interested in is how we might change the metabolic environment from a diabetic type state where there's too much glucose to one where we tell the body that it needs to change the way it behaves, for instance, using metabolic interventions that might well include intermittent fasting or caloric restriction. And so this program of work is being done in collaboration with colleagues in Oxford, in the Karolinska in Sweden, and in the University of Copenhagen. And we're starting to look at the effects of interventions through bariatric surgery, lifestyle, caloric reduction, exercise, and novel anti-diabetic drugs to see if we can impact on these inflammatory processes. And this is just a slide I put in at the end. This is work that we're doing in Oxford, looking at the effects of simulating fasting by giving beta-hydroxybutyrate, a ketone that's made by the liver in fasted states. And then finally, just to make the point that whilst I've shown you cells reprogrammed in the bone marrow, there might also be the opportunity to reprogram cells elsewhere in the body, in the heart, in the kidneys, and some of the effects that we see in diabetic pathologies in those organs might also be mediated by persistent epigenetic reprogramming. So let me stop there by acknowledging with gratitude the colleagues in my laboratory that actually done this work, in particular, Lauren Edgar and Naveed Akbar. But this is part of an ongoing and collaborative program, and our funders, the Novo Nordisk Foundation, British Heart Foundation, and the MRC. I'll pause, and I am also happy to take any questions.
Well, thank you very much, Professor Choudhury. We're privileged again to look into the laboratory of some world leaders in two areas that are of great interest to the issue of longevity and autophagy. And I'll leave Sir Christopher perhaps to open the discussion while I check on three questions that have come in on the Q&A, and I'll deal with those after you've raised whatever you want to raise, Sir Christopher.
Well, thank you very much, and thank you for that wonderful presentation, Professor Choudhury. You gave me glucose and genes for my letter G to take away with me. And I'm sitting looking at those two words and thinking it's a bit late to do anything about my own genes, at least until science gets perhaps a little further into the future. Blaming one's parents is, of course, a commonplace, but it doesn't seem to make any difference. As for glucose, you possibly could say more to the patient. I take your message. I should have moderated my glucose intake when I was younger, and doing it now seems to produce precious little difference. Is that all you can say to the elderly patient at risk of heart disease?
Well, I think you told us at the beginning that you're in your mid-80s. So I think you have your parents to thank for some very good genes, is the first thing that I would say. In terms of glucose management, it won't surprise you, it's quite complicated and we don't fully understand it. There may well be an effect of dietary glucose, carbohydrate, calories, which may be both directly metabolic, but also might be mediated by all those microorganisms that live inside our gut. And we're starting to scratch the surface as to how that might be involved in reprogramming our immune system, function of our liver, the function of our brain, and so forth. And that's a very exciting area that's still in development. But I think maintaining our overall, I frequently go back to ask, what could I do in the way that I live? How would I be most useful to my tribe? That being the thing that's defined me in evolutionary terms, and not eating too much and getting out and exercising and bringing things back to the tribe seemed to me like good starting points. I think, anyone that sits in the corner and eats too much and contributes too little is someone who's going to be pre-programmed to leave that tribal environment quite promptly. And people who are lean and active and don't eat too much are the people that your tribe wants to hang on to in terms of longevity. And I realize that's absurdly oversimplified, but that is there's a pair of very strong evolutionary pressures, I would submit.
Patients need it to be oversimplified, so I'm going to thank you for that. It's a wonderful message. And the surprising bit of it is bring something back that benefits the tribe, that acting in that way in our modern conditions might actually be good for our own health. What a wonderful message. I will take it on board. Thank you.
Thank you very much, Christopher, for that. Before I move on to Professor Sadoshima, I've just got two comments to make on that interaction between Sir Christopher and Professor Choudhury, which is, well, one reason we can't just blame our parents one way or the other is precisely what you talked about, Professor Choudhury, which is the epigenetic marking, which is us doing it. Do you want to comment on that before we go on?
Well, yes, you're right, and I mean there might be another layer of complexity in that our genetic makeup may predispose us to different types or intensities of epigenetic programming. But that accepted, it's certainly the case that the way that we conduct ourselves changes the way that we program these somatic cells. So yeah, I think we're learning more and more. And as a medical community, we've also not been blameless in this. We went through a period where we're not too concerned about carbohydrates, for instance, and we were obsessed with fat intake and isolation. And I think as we understand more, it's not as simple as that. And I think caloric restriction, particularly through carbohydrate, over excesses, rice, sugar, starch, pasta, bread, all of those things are useless calories to us that finish us off more quickly.
Well, I've got one other question for you, Professor Choudhury, and then two questions have come through for Professor Sadoshima. The question for you is, how can we normalize those macrophages? Does this mean that diabetics should have bone marrow transplants from healthy donors?
Okay, so I mean, clearly that would be quite an extreme intervention, but it's not ridiculous in my view. We don't have evidence that would help yet, but certainly we're certainly trying to figure out ways to reprogram the bone marrow without having to resort to bone marrow transplantation. But it's not science fiction that you might have an autologous bone marrow transplant. That means you self-transplant your own bone marrow in order to correct processes of homeostasis. At the moment, that's a long way off, and I'm certainly not advocating it. But other interventions that will correct the way that we manage our inflammatory status probably are accessible. And the example that I gave was fasting. So giving your cells a different metabolic stimulus apart from high glucose and seeing what they do with that.
Okay, thank you very much. I'll move on now to the two questions for you, Professor Sadoshima. Incidentally, both of them say thank you very much for a very interesting talk. And the first one refers to the fact that you mentioned that autophagy is primarily taking place during nighttime. Is there evidence that autophagy is connected to sleep or even specific sleep phases, especially given the important role of sleep in physiological maintenance?
So answer is, I don't know the answers. Sorry.
Oh, okay. Well, good to be straightforward and honest as a scientist, Professor Sadoshima. Okay, well, should we, excuse me, let's move on to the other question. Again, thank you very much for a very interesting talk. So my understanding is that unhealthy mitochondria undergo mitophagy. What happens to healthy mitochondria? I guess they divide and replicate, and could outcomes potentially improve in both heart failure and aging if we could increase the number of healthy mitochondria?
Yeah, so you are right. When mitophagy takes place, only a damaged portion of mitochondria is sequestrated by by fission. When mitochondria divide into two portions, good portion and bad portion. The bad portion goes to mitophagy, and the healthy portion remain alive, and also the mechanism to make new mitochondria is stimulated. So to keep condition of the mitochondria very good, I think mitophagy, stimulating mitophagy is very important. And also we have a mice which is lacking DRP1. It's a fission molecule. And if you knock out the DRP1 and then give high fat, surprisingly, those mice die within 3 weeks. So this is, we have been using high fat diet mouse model for a long period of time, but we have never seen such a drastic effect. So without fission mechanism, mitophagy mechanism, mitochondrial quality become really, really bad. So I think DRP1, maintaining the function DRP1 mitophagy is quite important for high-fat diet, obesity, high-fat diet, cardiomyopathy, as well as aging.
Yes, thank you very much. I wonder now whether we can see whether either of our two distinguished presenters have questions for each other. First, over to Professor Choudhury, whether you have any questions for Professor Sadoshima.
Well, I enjoyed your talk very much, and you alluded a couple of times to the effects of aging and diabetes on processes of autophagy, and I think you said that this is still very much under investigation. But I wondered if you could speculate, or where would you go with your first hypotheses, for instance, in how diabetes might impact on autophagy and related processes?
So yeah, I think overall idea is to how to reactivate autophagy even in the presence of severe metabolic condition. We know that too much nutrient, acetyl-CoA, very severely inhibit autophagy, and also DNA damage, reactive oxygen species eventually inhibit autophagy. So inventing how to reactivate this is a key, and that is common to metabolic syndrome as well as aging. So I really want to find a way to overcome downregulation mechanism.
And do you, Professor Sadoshima, have any points you want to raise on Professor Choudhury's talk?
So you raised maybe intermittent fasting, avoid glucose-induced epigenetic mechanism. But do you think that the type of food we intake, like a low carbohydrate, or would that be the key, or just reducing the whole entire calorie intake?
So I don't know is the answer, but I missed out some important parts of the story in the interests of simplicity and time. But one of the things that matters is that the epigenetic reprogramming of those cells is glycolysis dependent. Oh. So the cells have increased glucose provision, they take up more glucose, and this pushes them through a metabolic pathway of glycolysis, which is similar to the so-called Warburg pathway. So our thinking is that if we can switch those cells from glucose metabolism to fatty acid metabolism, for instance, then they will respond differently. But we're testing that. We haven't shown it yet. And of course, the other thing that's worth saying in the context of diabetes, most of the glucose in the blood is not coming from what you eat. It's made in the liver, which the audience may or may not know. So it's about a fundamental reprogramming of our metabolic pathways in order to give the cells something which is less harmful and potentially beneficial.
Yes, please carry on, Professor Sadoshima.
One more question.
Yes.
The fact that the body remembers you have had high glucose is kind of scary to me, but the memory lasts long. How long do you have to? How much do you have to take glucose for this epigenetic effect is activated, and how long does it last?
Yeah. So again, we don't know for sure. In the mice, we can. So in cells in culture, we can impose that effect in a matter of days. In mice who've been diabetic for several weeks, then the effects of those epigenetic modifications persist for at least 3 months. So we're looking at the moment in humans to ask exactly these questions. They're critical. One might imagine that given the nature of it, so your genes are in some sense immutable. And if you're going to fiddle with the way they're expressed, you ought to have a good reason for doing it. So if you take that as a first principle, one might imagine that the duration, frequency, intensity of hypoglycemia would affect the depth and resilience of the epigenetic reprogramming. That's our starting point. So if you've got a lot of hypoglycemia with big fluctuations over a long time, we think that will be deeply attached, as it were, modification. And the other thing that's interesting is, so Andrew Murphy and the Baker Institute in Melbourne has some very interesting data showing that transient intermittent hypoglycemia, so that's say it goes up a lot and it goes down a lot, but not in the diabetic range, is associated with more effects on driving the bone marrow to make new cells. So that the implication of that for humans is that even when we are in a pre-diabetic state, when we don't have the conventional criteria for diabetes, abnormalities in glucose handling might already be poisoning our bone marrow. And I think that is important. And I can see there are a couple of questions in the chat about public health interventions. And yeah, that's exactly where I am with this, is that we have to stop seeing this as categorical disease, you've got diabetes or not, and seeing this as a public health program of trying to keep people metabolically healthy for as long as possible.
So I'm a basic scientist, so I really like doing experiment using my own body, and nowadays you can monitor the glucose continuously. But do you think that we can get some knowledge from
Yes
measuring?
We're embarking on exactly that study. So we're interested in seeing whether patients with heart attack who have diabetes or not diabetes have an exaggerated inflammatory response and a depressed reparative response, and whether or not that's related to the absolute levels or the glucose fluctuation and so forth.
Yes, I've noticed, Professor Choudhury, that there are several other questions in the chat line, which I missed out on. One of them, if I can address it to either of you, are you saying that a body that is already physically, emotionally, and mentally stressed, the best way to heal is to restrict caloric intake?
Well, I'm not saying that exactly, but I'm saying that probably is at a population level, we eat too much. And so at a public health initiative, we should probably eat less. And those of us who are, sliding into middle age with, seeing the physical manifestations of that, probably ought to do some caloric restriction in order to regain metabolic health. I think that's what I'm saying.
Well, that leads to the other question here. Does your work indicate that governments should urgently tax or restrict the amount of high-fat, high-sugar processed foods aimed at children?
Well, I think, or educate. I mean, there are a number of ways of doing this, but I think in encouraging more metabolically conscious living for longer is definitely something that we should do. And I would choose to do it through education rather than taxation, because I suspect it's more enduring, but there might be multiple prongs. Okay,
we're near the end of our symposium. I want to find out, first of all, Sir Christopher, do you have any small snippet to add before we move to the closure? I usually tell a little story at the very end, and I will do that, but after giving you the floor first.
Thank you very much. I'm going to replace autophagy in my takeaway into second place, not only because it hasn't actually got an F in it, only the sound of the F, but because fasting has come through in both your presentations as very important. And therefore my F is going to be fasting, intermittent fasting. I think I can manage that, and you've talked about that. That's a very big takeaway message. Thank you very much for it. Thank you for your presentations. This has been a very worthwhile event for me, and I'm sure for others.
Well, thank you very much, Sir Christopher, for co-chairing it with me. I said I was going to tell a little story, and because we are at the moment in the midst of another major war, I thought I would just refer to the story of the Dutch starvation winter in the Second World War, where the Dutch children had to grow up with real malnutrition. And I'm afraid that that's going to happen possibly yet again in Europe, in the country we know about. But what is interesting about that fact is that not only the children inherited those metabolic changes, it's gone down 3 generations now, which means that those epigenetic marks that Professor Choudhury was referring to can actually last through from one generation to another. It's a highly controversial field in terms of whether these processes are well established, and it's not easy to say, but that's the best study I know of the transmission of metabolic disorder down through generations, prompted, I'm sorry to say, by a major war. And let's hope, for goodness sake, that that doesn't happen again. So with that, I think we round off this absolutely superb symposium with, we're privileged again to have 2 of the top researchers in the field, in particular in relation to the heart and cardiovascular problems. And we thank you both very much indeed. And we look forward to yet another symposium in a few months' time. So cheers to everybody. Thanks also to the audience for participating from all over the world. And we look forward to seeing you all again. Thank you.